Today we're going to be talking about incomplete and codominance. So let us begin. Starting off, our target for the day is really simple. We're going to be looking at crosses that involve incomplete and codominant alleles. And the goal is that you just understand what is going on in that inheritance. So let's look at a little bit of review. First, before we were talking about what's called complete dominance. And in complete dominance, it's really, straightforward. So if you happen to have, a red flower that's homozygous dominant for the red allele, then it's going to appear red. It will also appear red if it's heterozygous because the dominant red allele overshadows or masks the recessive allele. In this case, the recessive allele is blue, and the only way that the blue allele shows up phenotypically is if it's homozygous recessive. So the only way that you're going to have a blue flower is if it is homozygous recessive. Both the heterozygote and the homozygous dominant are going to appear red. If we were to go into a field and we were to look at these flowers as they were to breed, then what would occur? We'd start off, we have our parents, the parental generation, starting off with a homozygous dominant red flower, and we're going to cross it with a homozygous recessive blue flower. So we know genotypically that the only thing that the red flower has is the red allele, and we know genotypically that the blue flower only has the blue alleles. Okay? So if we cross them, we get to the F1 generation. And in the F1 generation, every single one of the offspring are going to appear red. Right? So when we go from the first generation, the parents, going to the next, the offspring, we'll see that every single one of them phenotypically appears red because every one of those offspring are going to be genotypically heterozygous, which means that they have both the dominant and the recessive allele. And since they contain the dominant allele, it masks the recessive allele. If we were to cross two of those offspring, then we get to what's called the F2 generation. And in the F2 generation, we would see something special happen. You'd end up with 3 phenotypically red flowers and 1 blue flower. So 75% of the offspring we would anticipate to appear red, and 25% of the offspring we would anticipate to appear blue. Genotypically, we would anticipate that 25% of the offspring would be homozygous dominant, 50% of the offspring would be heterozygous, and 25% of the offspring would be homozygous recessive. And that's the only one that appears blue. The other 3 would all appear red. In incomplete dominance, that changes. So for an incomplete dominance, you don't have a masking of one allele, but you actually have a blending of the 2 alleles. So instead of, say one allele hiding another allele, they both would cause a phenotypic change. So if they were for ears, and one is for long ears and the other is for short ears, you would end up with medium-length ears. If one is for a blue flower and one is for a yellow flower, they would mix and make a green flower. Okay? So that's what we're looking at when we're talking about incomplete dominance. So looking at those flowers that we just had in the previous example, we know that the homozygous for the red allele is going to appear red. We also know that homozygous for the blue allele is still going to appear blue. There's going to be no change in that. But the heterozygote is no longer going to appear red because these alleles are going to blend together. And since they're going to blend together, red and blue is going to give us violet. So now we have a violet flower. So the phenotype is actually revealing to us what the genotype is, because the red and the blue allele mixing together giving us the phenotype. So we can tell that it's heterozygous. So if we were to go to that same field and we took these flowers, we start off parent generation again. We take our red parent that's homozygous for red. It's the only way that we have a red parent. And then the blue parent, which is homozygous for blue. Again, the only way it can be blue, it has to be homozygous. If we were to cross them, then in the F1 generation, it would do exactly what you would expect. Every one of them would be heterozygous, which means 100% of the offspring would appear violet. So instead of having blue or red showing up in the next generation, they would all appear phenotypically violet because every one of them genotypically is going to contain both the blue and the red allele. And since they both have the blue and red allele, mixing together makes violet. If we were to cross two of these, then we would go to the F2 generation, and in the F2 generation, we are going to get, well, 25% would appear red, 50% would appear violet, and 25% would probably appear blue. Because we would see that 25% would be homozygous for the red allele, 50% would be heterozygous, having both the blue and the red allele, and 25% would have just the blue allele. They'd be homozygous for the blue allele. So that is how we would show or can see incomplete dominance. In codominance, it's different because they don't blend. Both alleles show up equitably. Right? So instead of mixing, you're going to have areas that show one of the alleles and another area that's going to show the other allele. So the trait is going to be expressed in different areas of that organism, but both alleles are going to be expressed. Okay? So it's not going to blend, it's not going to be a mixture, it's going to be different areas showing both. So instead of the flower that is blue mixing with yellow, making green, this would be blue and yellow spots or stripes or something on those lines. Instead of having long and short ears make medium ears, it would be like one long ear and one short ear, which would be really weird. But that's what we're looking at. So again, looking at our three flowers, we know homozygous for red, that's going to appear red. We know that homozygous for blue, that's going to appear blue. That's the only options that it has. But the heterozygote, that is going to appear where both red and blue are going to be expressed. In this case, it's going to be that the petals are both blue with like a red stripe or a red dot in it. Now, it could have been that it's every other petal just showed up as one color or the other. Right? There's different ways that this could have showed up, but in this case, it happens to look like there's a little sort of circle of red going around the petals. So if we were to go into our hypothetical field, again, taking a homozygous for red parent and a homozygous for blue parent, and we were to cross them, this time in the F1 generation, we would see, instead of the F1 being red or the F1 being violet, in this case, the F1 generation, they are all going to appear as the blue dotted red petals. So they have both the blue and red petals. 100% are going to appear that way because 100% of the offspring are going to have the genotype of being a heterozygote. Every one of them has both the blue and the red allele. And since they both have the blue and red allele, both are expressed, and we see the blue and red petals. If we were to cross two of these heterozygotes, then we would see phenotypically that in the F2 generation, 25% of our offspring would appear red, 50% of our offspring would appear blue and red, and 25% would appear blue. And that's because genotypically, 25% would have only the red allele. 50% would have both the blue and red alleles. They would be heterozygous. And 25% would just have the blue allele. They would be homozygous for that blue allele. Right? That's simple when you're looking at the difference between incomplete dominance and codominance. Incomplete, remember they mix, and codominant, one shows up and the other one shows up as well. They both show up equitably. To go and add into this, there's also a term called multiple allelism that we need to understand. And multiple allelism means that instead of having just 2 alleles per gene, we actually have way more than that. Some genes have dozens of different alleles. And that means that we have a lot more combinations than just the typical like red or blue, and that is the only options we have. So this is going to matter because now these alleles all have to interact with each other, and the way that they interact with each other is on a case-by-case basis. So one allele could be dominant to another allele, recessive to a second allele, codominant to a third allele, and incompletely dominant to a fourth allele. It could have all of these different interactions with different alleles. It just depends on which 2 alleles the organism has. Because remember, all these organisms that we're talking about are diploid. They are created through sexual reproduction, and in that process, that means that they get one allele from one parent and one allele from the other parent. And so the only thing we're concerned about is how each of those alleles interact with each other. So now going into this, instead of having just 2 alleles, the red and the blue allele, now we're going to add a third allele, the white allele. Now, blue and red, remember, we made them that they were dominant. And so in this case, what we're showing is that the red allele is the dominant allele. And how I'm showing that is, instead of just writing like 2 capital R's, I did a capital P for petal, and then a superscript capital R. And so this way I know that the capital P means that it is a dominant allele in all senses that I'm going to be concerned about right now. Remember again, our blue and red flower. Now the blue and red, it's going to have both the blue and red allele. It's heterozygous. And so that's why I did a capital P superscript R, capital P superscript B, so that way it can show that it has both dominant alleles that are codominant to each other. Blue flower, again, homozygous dominant, and in this case, capital P superscript B, capital P superscript B. Again, showing that it is a dominant allele in both cases. And then we have this white flower, and in the white flower, white, as it turns out, is recessive to both the red and to the blue. And since white is recessive to both the red and the blue, that means that I wanted to differentiate it, and I wanted to show it where I did a lowercase p with a superscript W. So that way we could see that it is a recessive allele. This also means that we don't just have one option for a genotype for a red flower. The red flower could also be heterozygous. It can have both the red allele and the white allele. The blue flower can also be heterozygous, showing both the blue allele and the white allele, or having both the blue allele and the white allele, because it will hide the other allele. It'll hide the white in both cases. Okay? So now we're going to take this and we're going to look at it in humans. In humans, we've probably experienced this a little bit with blood type. So blood type, we have 4 different blood types: A, B, AB, and O. Most of us are very familiar with those. Well, A blood type obviously has the A allele, and it can either be homozygous for the A allele, or it can be heterozygous with both the A allele and the O allele, because the O allele is recessive. B-type blood can also be homozygous for the B allele, but it can also be heterozygous with the B allele and the O allele. But if an individual has both the A and the B allele, if they're heterozygous with both the A and B, then they have type AB blood. And if they're homozygous, purely recessive with that O allele, then they have O-type blood. So O-type blood can only be homozygous recessive. A-type blood can be both homozygous for A or heterozygous with A and O. B-type blood can be homozygous for B or heterozygous with B and O. And AB blood can only be heterozygous with both A and B. We'll look more into this when we look at how multiple genes actually interact with each other, because most of you probably know about AB positive or O negative, and we'll get into that in the next lesson. Looking a little more into this, we're going to look at cows, because cows, well, their coat color is actually determined by multiple alleles, and those alleles are going to interact with each other in a special way. So you have 4 different alleles: black, brown, red, and white. Now, black is dominant to both white and red. Brown is also dominant to both white and red. Black and brown are codominant to each other. So a black and a brown, or a cow that has black and brown alleles, it's heterozygous for both of those, will appear black and brown. It'll actually be black with brown spots or brown with black spots, depending on how you want to look at it. A red cow, or a red allele, and a white allele are also codominant to each other. And when they come together, they make a color called roan. Roan is really special, and we'll look at that in a little bit. So in our first example, in the P generation, if you have a black cow that's homozygous for being black and a brown cow that's homozygous for being brown, and they were to mate together, you get the F1 generation, and you already know where this is going. You see this. It is, yes, they will all be black and brown spotted. Every single one of them, because every one of them genotypically has both the black and the brown allele. They are all heterozygous for those alleles. And if we were to cross 2 of those offspring, again, you end up with the F2 generation. And in our F2 generation, you already know it's going to probably be about 25% that would be black, 50% that would be black and brown spotted, and 25% that would be brown. The 25% that would be black would be homozygous for black. The 50% that would be black and brown would be heterozygous for black and brown. And the 25% that would be brown are going to be homozygous for brown. In another example, if we were to take, in our parental generation, a red cow and a white cow, well, the red cow has to be homozygous for red. It can't be heterozygous because red is a recessive allele. Same with white. The white cow must be homozygous for white. It can't be anything else. If we were to cross these 2 together, then we get these options. It could either look sort of tannish, or it could look pink. And this is where it gets a little interesting, because the tannish cow is actually not tan at all. It's a color called roan. And what roan actually means is that instead of being sort of big spots of fur, you have one hair that's red, and right next to that red hair, you have another hair that's white. And next to that would be a red hair, and a white hair, and a red hair. So this cow actually has both white and red hairs interspersed throughout all of its fur. And that's what gives it that coat color. So it would actually look one color from really far away, but as we get closer, we realize both alleles are being expressed equitably. And that is roan. So sorry, it would not look pink. We would end up with 100% all looking roan, because every single one of them would be heterozygous. They would have both the red and the white allele. So phenotypically, they would appear roan. Genotypically, again, heterozygous. And if we were to cross 2 roan cows, then we would anticipate in the F2 generation that 25% would probably appear red, 25% would probably appear white, and 50% would probably appear roan. That's because 25% would be homozygous for just the red allele, 50% would be heterozygous, having both the red and the white allele, and 25% would be homozygous for only the white allele. All right, our last example, guys, and here it is. Going a little bit more complicated. If we took a red cow, which must be homozygous for red, right, and a black and brown cow, so that must be heterozygous for black and brown. So again, the phenotype in this case immediately tells us the genotype. There's no other options for these. That's what they have to be. If we were to mix these together, if we were to cross these cows, we would get 50% of our offspring would appear black, and 50% of our offspring would appear brown. And every one of them would be heterozygous. Every one of our offspring would have the red allele in them masked by either the black or the brown allele. So the black cows, they're heterozygous with the black allele and the red allele, and the brown cows are also heterozygous, but in this case with the brown allele and the red allele. So if we were to cross one of our black cows with one of our brown cows now, what would happen? Well, let's look. In the F2, we would see that we get black and brown spotted, brown, black, or red. We had 25% for each different phenotype. So each one would show up equitably. The black and brown clearly is heterozygous for having both the black and the brown allele. The brown cow is heterozygous for the brown and red allele. The black cow is heterozygous for the black and red allele. And the red cow is homozygous for only the red allele. All right? So in conclusion, what we got is that the alleles, they don't only come in a dominant or recessive variety. There's different ways that they show up. You can have incomplete and you can also have a codominant interaction, right? And because of that, the phenotype sometimes actually shows us what the genotype is, because neither allele in this case is masked. They both are going to have an impact upon the phenotype, and that means we can tell what the genotype is in these instances. And remember, the inheritance patterns, look, they always stay the same. You get an allele from the female parent, and you get an allele from the male parent every single time. And because you get one allele from each parent, that means that these patterns are always going to follow with a very, very common sequence. All right? We can always track which way the alleles go, and sometimes, though, we're going to have more than just 2 options for a phenotype. Because in incomplete dominance and in codominance, you end up with where you could have 3 or maybe 4 different phenotypes. And it's possible, if we even look at more alleles that are present with multiple allelism, that you could have even more options. Okay? So we'll look more into that later on. And until then, be awesome, stay awesome.